Okay, recording. I'll share my screen. Is there anyone who can't see my screen?
Looks good.
Awesome. Thanks, Mike. So just like we have previously, if everyone could click on the small server on demand. I was still logged in because I just I stayed logged in. I just canceled my server. So some of you guys might have to log in again, which is fine. Again, same username and the password that you had changed into, and then you'll get your text message to confirm your identity. And then once you're here, small server on demand is a node that you want to pick, and then you can go ahead and click start. And as that's being pulled up, are there any other questions from any of the material that we went over last week? Anything you guys want to see again? Okay, that's all good. As questions come up, feel free to interrupt me, unmute your mic, put the question in the chat so we can get those answered. Again, I'm putting the link to the schedule in the chat. So yesterday we got through trim galore. So we finished trimming and filtering our FASTQ files. Remember, we filtered based on quality as well as to remove adapters, and then making sure that the length that remained of our reads was at least 20 base pairs long. So ended up getting rid of any read pairs that were less than 20 base pairs long, keeping in mind that if either the forward or the reverse read of an insert was less than 20, we had to get rid of both reads, because when you're working with paired end data, all the tools expect you to have both a forward and a reverse read for each one of your inserts. And so you have to make sure you have the same number as forward and reverse reads. And then I did just realize, so I'm going to start today with us actually taking a look at the trimmed FASTQ file, because I realized that we never actually opened one up to see what those look like. So we'll start with that today. So today we're gonna pick up where we left off last time. So now that we have our nice trimmed and cleaned up FASTQ files, we're gonna go ahead and do FASTQC again on those. And then we'll jump back into the lecture, and we'll talk about alignment, and then we'll go ahead and align our data in the FASTQ to counts Jupyter notebook, and then hopefully we'll be able to get through making counts tables. So we'll also do a strandedness lecture as well as that part of the Jupyter notebook. Everyone, I know I'm still waiting. Is anyone else still waiting for their instance to come up? Yeah, also waiting. Okay, we'll give that a little bit more time. So as we're waiting, since we're going to be going over these again, any questions from anything, any of like the quality that we look at in the FASTQC files? Any questions about why we trim or how trimming works? This is remember we use trim galore, which ultimately uses cut adapt under the hood. No questions? It looks like my instance is up. Yeah, Chief, of course. You can rejoin the meeting. Okay. Is there anyone that's still waiting for their Jupyter Hub instance to come up? Okay, great. So everyone's is up. So just to remind everyone where we are in our workflow here, so we've evaluated the quality using FASTQC of our raw reads, and then we went ahead and did some quality trimming as well as removed adapters using trim galore, and we're just about to assess the quality of those trim data. That's what we're going to pick up today. And so because we had some variables that we held last week, we're going to have to make sure that we still have those variables. So if you guys want to jump to the define paths to GL4U RNAseq Bootcamp process data and go ahead and run that code block. Again, Shift Enter on your keyboard, or you could press the play button up here. And I should also, too, have everyone check down here in the bottom left corner to make sure you guys are all running the Python Conda Environment GL4U kernel. Is there anyone who's not seeing this kernel in the bottom left? Okay, great. If that kernel ever goes away, or if you don't see it looking like that, please let me know so we can make sure that your kernel is working. So if you haven't done so already, go ahead and run this block to redefine all of the variables. Again, these are just paths to the scripts that we're submitting to the job scheduler, and also to the output files that are generated so we can all view them. And then we can actually use the table of contents here, and we're just going to jump down to number 2B, which is trim data QC with FastQC. So again, we ended last week kind of taking a look at these trimming reports from Trim Galore and seeing and answering some questions about the trim data. So some things you guys noticed was that there was more quality trimming that was done on the reverse read than the forward read, which made sense based on the FastQC analysis of those raw data. You guys also noticed that it actually doesn't remove any reads until it's gone through and done the quality and adapter trimming of both the forward and the reverse read. And again, that's to make sure that we always have the same number of forward and reverse reads, because we're working with paired end data. And we noticed that there was about 46,000, a little over 46,000 total reads that were removed, because after the quality trimming and the adapter trimming, that read length ended up just being too short, below that 20 base pair cutoff, and so those were ultimately removed from our data. And so before we get going here, I do want to show you guys what this trimmed FastQ file looks like. So first thing I want to do is just make sure that I have that. We should. So if everyone wants to remember the command that we use to list what's in a directory, you guys type it in the chat. Yeah, you guys got it. So exclamation point, again, because we're in a Python kernel, we run a Unix command. And then we're going to use the variable that we defined up at the top that holds all of the trimmed FastQ files. So I'm just going to put this command in the chat. And then if you guys all want to run that with me, I do have one sample in here, that flight rep1 sample. So the first biological replicate of our flight group. And we see that there's R1, so our forward read, and R2, our reverse read. And so a couple things I'm going to do here. Let's add a couple more blocks. And I just want to take a look at the beginning of both of these so you guys can see what they look like. So does anyone remember the command that we use to open up a file and look at the contents? You guys can go ahead and type it in the chat. Yeah, so cat is it, but this is a little interesting because cat will open up a file as long as it's not compressed. And you'll notice that these files ending in .gz, they're actually gzipped compressed. And so there's kind of like a sister command to cat called zcat. And what that does is it will open up and allow you to look at a gzipped file, which is pretty convenient. But keep in mind, these files, as we noticed from the FastQC analysis, at least of the raw data, and we only removed 47,000 reads, they have almost they have millions and millions of reads in here, and each read is 4 lines, as we know, and so it has like a lot of lines in this file. So if we were to just zcat and open up this whole file, there would be a ton in there. So we can use this pipe function, and that will push the output of this command into a new command. So what's the command that we would want to use if we just want to view the first few lines of a file? You guys remember the command that we use just to look at the first few lines of a file? -n is an option for that command, but what's the actual command? Yeah, head, yeah, exactly. And so we're gonna run zcat, but then we're gonna pipe this output into head just to see the first 10 lines of the file. And we shouldn't have to add an exclamation point here because we added an exclamation point at the beginning. So I think that we don't have to here at this point. So when you just run it like this, what head will do is it will take the output from this command, this zcat command, and pipe it into the head function. And so this is going to start unzipping and allowing us to view the contents of this trimmed FASTQ file, but it's going to stop after 10 lines because we told it to go into the head function. So we can all run that command, and you should see something that looks like this. Is there anyone who's not seeing this? Let me put this command in the chat. And so what you'll notice, it doesn't continue off the page. So anywhere there's a line break, it'll just move it to the next line. But you'll notice here, how many reads are we seeing right now? Let's put it in the chat. How many reads are we looking at? Yeah, 2.5, Joel, exactly. So remember, the head command will give us the first 10 lines of a file unless we give it an option like that -n option that Mark pointed out in the chat. So we didn't give it a -n option, so it'll default to its standard, which is 10 lines. And we know that each read is 4 lines, so it should show us 2 complete reads and then half of a third read. And so that's what we're seeing here. So remember, all of our reads start with this @ symbol, followed by information about the flow cell, the actual cluster that it's in within the flow cell, and then we have some information here that this is the first read of the sequence, and then we have the, we also have here the index that was used for this. And so this is actually single index. And then the second line is our sequence, our A, T, C's, and G's, so our sequence of interest here. And then our third line is our spacer line that usually has a plus sign. And then our fourth line is quality scores that correspond to each one of the base calls made. And so that's one read. And now we have a second read starting here, again, starting with that @ symbol. And then we have our ATCs and Gs, our spacer line, and then our quality score line. And then we have that third read, but just the first 2 lines of that third read. Another thing I want to point out is you'll notice that one of the pieces of information in this header, again, so long as the header has not been tampered with, is cluster location. So I think that this area, I want to say here, is the cluster location. So you'll notice that it's different for each one of these reads, right? Because that read came from its own cluster location or its own little dimple on the flow cell. So it should be a unique address. And the other thing I want to point out this is now when we look at the reverse reads, because each one of these inserts are read in one direction, the forward direction, and then the reverse direction. And so because the forward and the reverse reads are from the same cluster, we should see these headers match up in the same order that we see them in the forward. So let's now run this same command again, but this time we're going to look at the reverse read for this sample instead of the forward read. So instead of that R1, we're going to look at the R2 file. I'll put this in the chat. So now if we go ahead and run that one, you should see the same thing that we found before. But of course, the sequence is going to be slightly different, right? Because it's in the reverse direction of that same insert. And another thing I just wanted to point out, and how we know that these are mate pairs with each other, is you'll see that, right, so this first read in our forward file, we see the same exact header information, which is basically an address to where it was on the flow cell, is going to be the same exact thing as the address to its respective reverse read. So you see that 11261 value here. All this is the same flow cell information. So this is in the same cluster, except a couple of difference, right? Whereas this is 1 telling us that it was the forward read. This is going to be 2 telling us that it's the reverse read. And then you'll also notice, because these are all from the same sample, it's not, which one isn't running for you, Mark?
The one you just posted right now, for some reason it's not running. It says while our command is running, let's view an example of the standard output from this command, but it's not showing anything.
Did you copy and paste this one? So I added a cell block here.
Yeah, I did copy and paste this one.
Do you see that you have your Python kernel running in the bottom left?
Uh, yes, Python conda NVGL4U. Oh, it says busy. Didn't say idle.
Busy means that it's running.
Oh, okay. So it's just taking a while to load up.
Yeah, once it
Oh,
finishes, oh, go ahead.
I said, oh, okay.
Once it
That's
finishes, it
probably
should populate with a number in brackets. The other thing is did you pipe it into head?
pipe it into head. Yeah. Yeah, I have head on there too because I just copy paste this whole thing, right?
Yeah.
Yeah, I did. It's still running. I just saw it says memory 618.18 out of 4096 megabytes, so I think it's just downloading everything.
Hmm. Should go
That's it.
pretty
Yeah.
quick. Is anyone else held up looking at this? Mark, I'm
So this is Elba. I just basically start a new kernel every time I open up and just run all the code, and that clears all the problems as we did the first day.
Okay.
That's
Maybe I
that
should.
works for me. I tried doing it again today. I've done that several times today. I said, well, let me try because the page looks like how I left it, but now I'm just running everything again.
All
So I
right.
would look for directories. They weren't there. That's the clue when you do a, print working directory and you're in some weird, doesn't give you what you need. So I just find starting fresh with that kernel and new parameters helps. I don't know if that's the issue you're having though.
No, it seems like everything's running a lot slower than normal. So
Mm.
maybe I should just restart the kernel and re-put in the variables and just try it again.
yeah.
Yeah,
All right, I'll
if
try
it
that.
takes so little time to do it, I find that maybe I'm just gonna do that always.
Yeah,
Just to
Okay.
IT
move
support.
along.
Turn it off, turn it back on again.
All right. Thank you.
Yeah, give that a try, Mark. And then if restarting, you can either like refresh your browser or you can come to the kernel here and then you can select the Python Conda environment GL4U and click select, and then that should have it go again.
All right.
Give that a try. And then if you're still having issues, let me know. In the meantime, I'm going to finish doing this comparison and then touch back to make sure that you got it.
Thank you.
No problem. Yeah, so Mark, take a look at my screen while you're waiting for yours to refresh. And so here, as I was saying, is that the forward read and the reverse read, these reads should be in the same order for each respective insert that it's sequencing. So again, in the forward direction, the reverse direction. So that's why we see this kind of address to the cluster that's here in the forward read is the same address, right? Because it's the same cluster in the reverse read. And then again, the difference being here we have a 1 telling it this is the forward read, 2, this is the reverse read. And then you'll also notice, since this is all our flight rep 1 sample, and then keep in mind those indexes that we add to the adapters are going to be the same index for each sample. That's how we know which sequences came from which sample. So you notice this GGA is going to be the same in all reads for both forward and reverse, because again, that they're both from the same sample, this flight rep 1 sample here. And then you'll notice that the next read down, if you take a look at that read address in the forward read, you should see that it matches exactly the read address. Awesome. Thanks, Mark, letting us know. You'll notice that that second read has the same read address as in the reverse. And so again, that's how you know, and that's what these tools are looking for, is these the forward and the reverse read coming in the same order in the file and also having these same addresses. And so now that we have successfully trimmed our reads, we're going to, and you guys probably should see this, geez, it broken pipe. That's just because we piped it into head, so it stopped unzipping the file. Nothing to worry about. And so the next part that we're going to do now that we trimmed our reads is we're going to assess the quality of the trim data now, again, using FastQC. So the same tool that we use to look at the quality of our raw reads, we're going to use to look at the quality of our trimmed reads now. And so just like we did with the raw reads, the FastQC does take several minutes to run. So instead of waiting for everyone's to run, we're going to submit these jobs to a job scheduler. Before we do that, we'll go over the commands. So again, this should look familiar from the raw data. So again, we call FastQC using !FastQC because we're running in a Python kernel. Dash O, does anyone remember? Well, I guess the answer is here. The dash O parameter is the location for where we want to output the FastQC output files. And so we're outputting them into the directory organization that we had put together last week. This time in the O1 TG, standing for trim galore, preprog standing for preproc directory, and then the FastQC report subdirectory from there. And then this last part here is going to be a positional argument to indicate the reads that we want the FastQC program to work on. So this now is going to be the FastQ files that have been trimmed. And again, same setup as before. We have this sample variable that we're calling again in the curly brackets because we're going to run this in an array script. So that sample variable is going to change based on which sample it's working on. And then R in this wildcard here, so that way it does both the forward and the reverse. And this time we've actually renamed the files as part of the script to end in trim.fastq.gz. And so now that we know all the parameter definitions, let's go ahead and submit that job to the job scheduler. So kind of scroll past those additional blocks that I had you guys put in. And we're looking for this sbatch command. Again, sbatch is to tell the program that we want to submit this to the Slurm job scheduler. And then processing is just the variable that holds the path to where all the processing scripts are. And this is the processing script for our trim data. And then the name of the script that I had made is trimmed.fastqc.slurm, and that's the script that we just went over. So if you guys all want to go ahead and run that, you should get an output that says that your job was submitted, again with the job ID here. And because of the cooking show magic, we won't have to wait for that to be completed. So we're going to view an example of the standard output from this command. So this should look very similar to you guys. So if you guys want to go ahead and run this cat command. So this is the standard output for the file. So again, it's what's reported from the file as like a progress log. So this happens in addition to whatever output files the tool generates. So this should look familiar from the raw one. And again, this header stuff here is all those unique things that I add to the scripts, just to echo and tell me what job it is. So here I have the job being GLDS 104 because that's the data set that we're working on, trimmed FastQC. This one was the wrong one that I just echo out. I always do a timestamp to print out the start time, the host name, so the server that it actually runs on, the array task ID to make sure that it's the number that I expect it to be, the name of the sample that it's working on, and the version of the tool that I'm running, in this case FastQC. And then I have it print out the exact command that's running just to make sure that those variables that are put in place are indeed what I expect them to be. So this should be flight rep 1 held in that sample variable here as we see. And then also we have the forward and the reverse read that we're going to be running this program on. And so just like what we saw with the raw data, we again see this log that comes out of the FastQC analysis. So every 5%, it kind of gives you an update. And it goes through and does the forward read first, and then it did the reverse read second. And so what we're really looking for in the standard output is to make sure that we see these lines here, that the analysis was complete, this being for the forward read, and then down here we have analysis complete for the reverse read. And so we also want to check this to make sure we got no errors. So we want to make sure that both analyses were completed and that we didn't end up with an error anywhere in our standard output, which we didn't, which is great. And then I always have at the end of the scripts that I write, always have a timestamp at the end right here, just a series of commands to print out the total time in seconds, minutes, and hours. So you'll notice that this took 15 minutes to run. And so rather than taking a look at each sample individually for the FastQC analysis, as you guys remember with the raw data, we were able to compile all of the FastQC results for all of our samples into a nice little comprehensive report using the multi-QC tool. And so we're going to use that same tool to compile all of the FastQC reports from our trimmed reads now. And so again, this command should look familiar here. So we have call it with multi-QC, -N for the multi-QC tool is telling you, you're going to tell the tool what you want the prefix name to be. So this time I want it to be trimmed _ multi-QC because this is a multi-QC of the trim data. -O is indicating the output directory to store the multi-QC results. So this is going to be set up right in our directory structure that we had built, except this time a subdirectory under FastQC for the trim data, for the trimmed multi-QC report. And then as a positional argument, this last thing here wants the directory that holds all of the FastQC files. And so we can go ahead and run this command. And this one we're running, it's really quick, just like you guys remember for the raw data. So we're all running it right on the screen. So we're seeing the standard multi-QC output being generated in real time. And so similar, is there anyone still waiting for their multi-QC command to finish? Okay, so everyone sees what I see? And just a reminder of what the standard output for multi-QC looks like. This tool is nice. It tells you what version it is. It also lets you know if there's a newer version available. So we see here there is a newer version available. It tells you the path that it's looking at. Again, this is the path that we provided as a positional argument at the end of the command. So it's looking in this directory. It looks for anything that the multi-QC tool can consider an output that it recognizes to compile. So honestly, I'm not sure what this snippy is or what looks enough like it. But we're really looking here for anything that it found that's FastQC related. And so it found 24 reports. Is that what we expect? Yes, no, no, yes. Yeah, it is what we report. Why, Jill? Why do we want 24? You can unmute your mic. Yeah, 12 forward and 12 reverse reads because we have 12 samples in total, 6 flight and 6 ground control. So 24 reports is what we expected to find, so that looks good. Then it just gives you some log information that's compressing the plot data, generating the HTML, and then putting all of the files into its output directory. And it'll tell you at the end here that multi-QC is complete. So again, when looking at the standard output for multi-QC, we're really looking to make sure that it finds all the reports we expect for the tool that we're looking at for it to compile on, which is FastQC. We want to make sure that it says that it's complete, and we want to make sure that we don't see any errors in the standard out. So all things that we're looking for there. And then just as a little reminder here, in addition to parameter definitions, I always have the input data, right? So it needs that fastqc.zip or all of the fastqc data from all of the samples that you want to compile. And then the output data that it gives, similar to the raw fastqc analysis, it'll give us a directory called trim multi-QC data. And then it'll also give us those data plotted in these nice comprehensive plots in the HTML format. So we're looking for those two output files, and then we're going to view the HTML report and answer some questions about the trim data. So in order to view HTML reports in Python, we have to run this IPython display from iframe. So everyone go ahead and run that code block. And once you see that that's completed, now you should be able to view the trimmed multi-QC HTML report by running the next block through iframe. Is there anyone who wasn't able to pull up their multi-QC report? Okay, great. So here, again, should look familiar from the raw data. Keep in mind you could move this back and forth, up and down to go through the data. If you want to change what it looks like, how big, how much information is displayed without scrolling, you can always modify these width and height settings in that iframe command to display this differently if you'd like. So it tells you what the report was generated on, and then it gives you a summary of everything for each sample. Keeping in mind now this is the trim data, and so we see each sample name here followed by R1 or R2 indicating forward and reverse. Again, it summarizes the % duplications. Keep in mind duplications we always take with a grain of salt with the FastQC because it's really subsetting the data when it looks at that percent GC content and then the total sequences in millions. And keep in mind that all of these columns here are interactive, so you can toggle them to sort it based on the highest value for that column or the lowest value for that column, which is going to be useful in answering the questions. And so we see a summary of all the data up there. And then as you scroll through, you're going to be able to see similar data to what's in the summary. So here we have unique reads and duplicated reads. And the thing I always like about this too is you could do the raw numbers, or you can view it as a percent, which makes it easier to compare the different samples. And then as you continue to scroll down, you'll see the histograms showing the mean quality scores at each position. Again, position there on the x-axis, and your red quality score on the y-axis. The higher the Q score, the better the quality. And again, if you hover over these lines, it'll tell you which sample that you're looking at. And as we continue to scroll here, this is another way to look at the quality scores. This time you're looking at the quality scores based on count. So you got the average quality scores on the x-axis here, the count on the y-axis here, and you can hover over to see the value for each individual sample. Seeing really good quality here overall. Per-base sequence content, so some things are a little interesting here. So this is looking at the percentage of ATCs and Gs at each position of the read. And again, the different colors here indicate that wobble at the beginning that is unique to RNA-seq data because of the hexamer random priming that we do to convert RNA into cDNA. So this we usually see. A couple of interesting things we're also seeing here. Here at this position, which if you look here, it tells you the position. So it tells you the position up here that you're at, and then it tells you the actual percentages, and each line here, each row is going to be a different sample. And so kind of one thing that we're noticing, even just from the color-coded, we have something weird that's happening here at position around 100. And then we have a similar thing that's kind of happening over here that's pretty weird. So you'll notice that like that percent A is pretty much 0 in these locations. Any idea why? I don't have a question about this. So this was something that we actually talked about when we were doing a dry run of this.
Can you repeat the question?
Yeah, so you'll notice here in this plot that looks at the percentage of ATCs and Gs at particular positions across the read. It looks like at 2 locations around 100 base pairs, where my cursor is over now, and also at around 150 base pairs, we see that at those 2 locations, the percent As are really low. So here and here. And I'm just curious why you think that could be. Was that the initial stop point? What do you mean, Joel?
I mean, didn't you send it off once and only get it sequenced out to 100 initially? I'm just spitballing ideas.
Yeah, so that's part of it, right? So those locations being at around 100 and 150 makes sense because we know that we have 2 different size reads that we started with, right? We started with reads that were either 100 base pairs max or 150 base pairs max. And I told you that was a caveat of us outsourcing the sequencing and then giving us sequences at 2 different reads. So that's part of it, but why would there not be A's at those locations? So if we're doing quality trimming and adapter trimming. So quality trimming happens first, adapter trimming happens after quality trimming. So the last thing that we trim for are adapters, a Illumina universal adapter.
And so the universal adapter starts with A, I'm assuming?
Exactly, yeah.
There we go, okay.
Yeah, that's exactly right. So the Illumina universal adapter, should I have it up here or not? I think I do in the trimming command. So if you look at our trimming command, right, we have the universal Illumina adapter listed here, and it starts with an A. And so this is the first thing that it's looking for when it does the adapter trimming and we have it set really stringent to where if it identifies 1 base pair that matches the universal adapter, it'll cut it off. And so it's stringent to 1 base pair, which means that if it found an A, at that 3' end, it's going to cut it off, assuming it's an adapter. And so the caveat of that being that after we trim at that end position, we're never going to see an A because it'll trim it off. So anytime you see something like weird patterns like that in the data, it's always good to kind of think about what's going on and what's happening that might cause it to make sure it's no issue that like you did or no issue that could have happened in the wet lab. And so just wanted to point that out. It was something that had come up, and honestly, I had never really thought about it until it was brought up. And Mike on the line actually helped identify that answer of why we're seeing that. Next we have here is our per sequence GC content. So this is the average GC across reads. And so this is still kind of looking pretty similar to what it was for the raw data. So remember that Rachel did a blast search on some of those overrepresented sequences that she saw in that first sample and identified them as ribosomal. So ribosomal RNA can have some highly repetitive sequences, and we noticed from looking at those sequences that they had a lot of C's and G's in there. So this peak is likely that ribosomal contamination, and that's not something that we trimmed for. So we don't remove any of that, so it makes sense that that's still there. The other thing that I point out that this could account for is in the NovaSeq or in the MySeq. I think this was sequenced on a MySeq. It does 2-channel. So I know I always say that the A, T, C, and G is each their own fluorophore. In order to speed things up, right? Because if you have those 4 wavelengths, then you have to take a picture in each one of those wavelengths. So to speed things up, some of Illumina's sequencers uses different chemistry for the base call. And so the one that this was sequenced on actually uses 2-channel chemistry. So it looks like this middle block here, I don't know if you guys can see that, make that bigger. So this 2-channel chemistry here, so rather than having the A, T, C's, and G's each have their own unique wavelength and having to take 4 images to save time, what Illumina does is does a 2-channel chemistry, and some of the machines, they even do a 1-channel chemistry. Actually, the iSeq does 1 channel. But in the 2-channel chemistry that these data were in, we have an A, which is linked to 2 different fluorophores in green and red. So that'll show up if you take an image in the green wavelength as well as the red wavelength, will tell you an A. G has none, so it'll actually be blank for both those images. T just has green and C just has red. And so that's how it's able to distinguish the base pairs. The issue with that is if you're sequencing along, right, in your base pairs and everything's looking good, right, so you know that that spot has a sequence there, has an insert there that you're sequencing. If that polymerase falls off early before it's done sequencing the rest of that insert, which does happen, when you're taking the images at 1 and 2 and there's no polymerase there, which means no more sequencing is happening, it'll be blank, right, for wherever that polymerase falls off. But it'll assume that that's a G, because G doesn't have any fluorophores attached to it. So that's also sometimes towards the end of reads. If the polymerase falls off early, you can get a string of Gs, and that's just a caveat of doing the 2-channel chemistry. There actually is an option in the trim galore cutadapt parameters that account for that. It basically doesn't consider the quality score of a G base call in doing the trimming. We actually don't do that at GeneLab, but it's something that we might want to consider. And the reason that we don't do that is because we, of course, ingest sequence data from all over. It's not just the ones that we produce in-house. And so there are other Illumina machines that do the 4-channel chemistry. So we don't want to be biased in that manner. So that's
Rachel, quick question. How do you distinguish if you basically say, if I see no signal, you're a G? How do you distinguish that from I can't read you?
You
Right?
can't. Yeah, so if it happens early on, if there's no insert that's right, so if you have a one of your little dimples, one of your little cluster regions on your flow cell that no insert latched onto, it's going to be dark from the start, and so that it'll just assume that there was nothing in that cluster, right? But the issue is when there is an insert there, but the polymerase falls off early. And so it can't distinguish. Once it falls off, it'll always read it as blank for both the image pictures, and it'll assume that it's a G, and it'll also tell you that it's a high quality G. And so, yeah, that's kind of just a caveat of the chemistry here. So that also sometimes when you see when you see this little GC blips, it could be some of that too.
So and I don't want to bog the group down, but I'm really interested in technology. So it basically is making the base calls and fluorescence based to relative to a baseline image it takes at the beginning.
Right. Yeah. Yeah, so that's kind of
So the G will give you something above threshold, but just not the fluorescence. It's again.
Yeah, pretty much, right? So, there's always going to be the background, right? So that you take away, but that background signal is going to be there because you have all these ATCs and Gs there in your flow cell. And so the ones that latch on and release the signal, again, if it's a G in the 2-channel chemistry, there is no fluorophore attached to the G. And so it's always going to be blank for both of those images. So if it wasn't blank, if it's blank at the start, then it knows that, okay, there's just nothing in this cluster. But if there is something in the cluster but is an insert that it's sequencing, and if that polymerase falls off early, then it'll assume that it didn't fall off and that blank is just being read as G's. And so that's
Okay, so if it reads a blank between 2 base calls, it's a G because there was
Right.
something after it.
Yes.
Okay.
Mhm.
Or when it falls off, the last
When
thing,
it
whatever.
falls off.
I'm sort of getting the
Yes,
sense of how this works.
exactly.
So,
Right.
okay.
Yeah, so kind of like a caveat of the chemistry here. And of course, Illumina sequencing is really good. So it doesn't happen like often, but it does happen. And so sometimes you'll get a little blip in the GC content when you do your FastQC analysis. And it could be a contaminant such as the ribosomal RNA that Rachel identified last week. But there is a chance that it could also be just kind of a string of Gs because of that caveat of the 2-channel chemistry if it was sequenced on a 2-channel platform. So just things to keep in mind to understand the technology and limitations of it. But of course, so long as that string of G's along with whatever came before it, the sequence that came before it, doesn't match to something on your reference genome, it shouldn't impact your data. It probably just won't be aligned depending on the settings that you use for your alignment. Next graph here is the per base end content. So keep in mind, we're only trimming from the 3' end, and we didn't really see too many ends at the beginning or in the raw data. So of course, we're not seeing too many ends here. We do still see that blip at the 5' end because again, we don't cut at the 5' end. We don't trim at the 5' end. Some people do. And again, this is why we kind of looked for what would be consistent for all of the organisms that we have, for the variety of different RNA-seq experiments that we have, and kind of came up with the consensus pipeline. And people feel strongly about trimming or not, so we wanted to be as conservative as our trimming as we can and really only trim for quality and adapter. But there are some people that are aggressive with their trimming, and they will trim from the 5' end too, so you can do that as well. I see that Ty just joined. So Ty, just a quick summary. If you could log on to your Jupyter Hub instance. Let me go ahead and put that link in the chat again. So I put the link in the chat, and then just make sure that you click the small server on demand for your server instance. And then once your server is up and running, let us know. And then I'll have you join a breakout room with Lauren. And then Lauren, if you could get Ty caught up with the FastQC.
Sounds good.
Awesome. Yeah, so just shoot a message in the chat when your instance is up, and then I'll have you hop with Lauren to go through this FastQC real quickly, and then you'll join us again, and we'll be at the start of the alignment lecture probably, or still answering questions. Sequence length distribution. This probably looks a little different than it does in the raw data. So if you remember, the raw data had a very clean, all of them were overlapping sharp peaks. This you kind of see a little bit more variation. Again, hovering over, you can see the different samples. Sequence duplication levels, again, taking this with a grain of salt. We didn't CHOP for any ribosomal RNA contamination, and what this might look a little bit better, and I think it does look a little better than the raw data, just if there was any sequences that were mainly adapters, if we had any adapter diners in there that would have been a repetitive duplicate sequence, those would have been removed with the adapter trimming. Overrepresented sequences. So again, this just gives you a summary in the graph. The unfortunate thing about multi-QC and compiling the FastQC reports is that it doesn't actually give you the sequence information. And so here, if you just notice that anything had like a big percentage of sequences that were overrepresented in one sample, or a handful of samples, then you might want to go back to that sample's FastQC results and take a look at those overrepresented sequences, because then you could do a blast search on them to try to get a better idea for what those overrepresented sequences are. Adapter content. So whereas before we had a graph that showed all of the samples and the adapters that existed in them, and here you'll notice that what multi-QC does is that if it doesn't identify any adapters, it won't even graph it. It'll just let you know that no samples were found with any adapter contamination. So this is kind of giving away one of the answers to the questions, but taking a look at this, we see that we have successfully removed all of our adapters. And then again, the multi-QC at the end kind of gives you a summary of each one of the analyses that's done on the data. And so I want you guys to take a look at that multi-QC report and start answering some questions. Before I get into the questions, I do have, I am curious how you guys are liking the Mentimeter questionnaire. Do you like that or the polls better? The reason that we switched to the Mentimeter one is because it allows us to display all of the answers that all you guys gave without associating it with a name, so you can kind of get an idea of what everyone else is thinking. So I don't know if you guys like that better, if you'd like us to go back to the poll.
Makes no difference to me.
Okay. Yeah, good, doesn't really matter. Okay, we'll do the Mentimeter then. So if, I don't know, Armin or Lisa, if you guys want to put the links to...
Yep. So we'll post that right now.
And this is probably one of them that had to be split up.
I got it.
Questions.
I just sent it.
Oh, you did. Okay.
Great. Thanks, Lisa. And I think, is the answer key still the same one that I had used last time?
Should be.
Okay, I still have that up, so. So after trimming and filtering, did any read pair, or was that the one that one?
No, we should be on how many reads are there in the GC.
Yeah.
So you guys should all actually go to.
Awesome. Ty, if you want to go to the breakout session and go to Lauren's breakout room with her, and then Lauren, if you could just walk Ty through the FastQC.
Sorry.
Sounds good. I'll see you there, Ty.
Sorry, the code should be 2818 at the end.
I think the link should work. I just didn't present the right question.
Oh, okay.
Try the first link again. There you go.
Yeah, if you hit refresh to the link that Lisa put in the chat, it should work. Or if you use the link that Armin sent, you'll have to type in the code. Armin, I don't know if you want to correct the code in case people go that route.
Yeah.
So go ahead, type your answer, and then hit submit. For how many reads are there in ground control rep 1 sample after trimming? What about flight rep 2? And are they still the samples that were sequenced at the greatest and least read depth respectively? And I have a little hint here for what section of the multi-QC report for you guys to take a look at. And so Lisa or Armin, if you guys could just give me a head count for how many answers you're seeing.
Yep, we just got one so far.
Yeah, 15 participants, so we're waiting for 15 answers. I probably won't answer, so 14 answers if all of the instructors answer as well. go ahead, take a look at the multi-QC report for our trim data under the end seats in the general statistics section at the top is where you should be able to find the answer to this question. How many answers we got?
We have 5 so far.
Give you guys a little bit more time.
We have 9 now, which is the number of students.
Yeah, as long as no instructors answered. I'll give another 10 seconds and then we'll take a look at the answers together. Okay, I'm gonna go ahead, take a look at these answers. Let's see that. Here we go. Here we go. So let's see. A lot of 85.6, 65.2, there's 68.3. Okay. A lot of 68.3 and 60. I hope. Okay, let's take a look at this. So we want to know after trimming, we want to look at ground control rep 1 and flight rep 2. And we want to know what the total number of reads that are left in each one of those samples after trimming, and if they're still the samples that have the most and the least number of reads respectively. So let's go up. So the section we want to go to is under general statistics, so right at the top of the multi-QC report. And then we want to know ground control rep 1, the number of reads that remain. And so if you take a look here, ground control rep 1, you just want to go over to this column here, which is the total sequences in millions. So those of you guys who said 85.6, you are correct. 85.6 million reads is how many reads we have in ground control rep 1. And of course, the number of reads in the forward and the reverse for that sample should match, and they do. And then I also asked you to look at flight rep 2. And so you can scroll down or you can toggle on that if you want. So flight rep 2, I have down here. And again, if you scroll to the right under the millions of reads, there's 65.2 million reads for flight rep 2. And so that answers that question of what are the total number of reads. For those of you guys who didn't get those answers correct, do you still have any questions on how to find those answers? Okay, so clear now? Feeling more comfortable reading this? The second part of that question was, are they still the samples with the most and the least total reads respectively? And to do that, I don't think many people answered that in the chat. I think I just wrote the numbers, which is fine. But you can use this to toggle, and so to sort, I should say. And so I just sorted this with the highest number of reads on top, and we'll see the highest number of reads, that 85.6, does still belong to ground control rep 1. And then just clicking it again, sorting it in descending order, or I should say in ascending order. So with the smallest on top is 65.2 million reads, which is indeed the flight rep 2 sample. So they are still the samples with the most and least read depth. Questions about how to view and take a look at number of reads and samples on a multi-QC/fast QC report? Okay, good. I'm going to have you guys look at this a little more for the rest of these questions to get you a little bit more comfortable navigating and understanding the information that's in this multi-QC fast QC report. Second question, if Armin and Lisa, if you guys want to change to the next question, which is, what is the sequencing depth range among samples, and is this an issue? So what is the range of sequencing depth after trimming? Related to the first question. Give you guys a few seconds to answer that. Lisa or Amr, if you guys just let me know how many people responded.
No one yet. Oh, one. We have one.
Yeah, so kind of the answer to this is in the answer to the first one, the sequencing depth range. So what's the range of sequencing depth or number of reads? The highest and the lowest in the range. And is this going to be an issue for us? We touched on this a little bit last week when we went over the raw data.
If you guys aren't seeing the question that Amanda has highlighted on the screen, you should just hit refresh.
How many answers do we have now?
3.
I'll give you guys about 30 more seconds. It's pretty straightforward. It's not a trick question. It's obvious from the first answer. How many answers do we have, Lisa?
7. So I think we're waiting for a couple more. I think 9's a good number. Last time after 9, we didn't really have any more answers.
What's the sequencing depth range? We have 10 more seconds for the few of you who haven't answered yet. You guys put in your answers, or you put I don't know if you're unsure. Okay. Let's see. $20.4, 6.85.6, same as before. Yeah. Yeah, everyone who wrote this is correct, right? So it's 65.2 to 85.6, right? So the range is the least depth sequence and the most sequence. And so we know that because we just looked at which sample was sequenced at the most and least depth. And so we know it ranges from 65.2 to 85.6. So we can have a difference of up to 20.4 million reads among samples. And is this an issue? Well, it could be primarily maybe for lower expressing genes, but we will do a normalization step before we do differential expression analysis to account for differences in read depth. So where this would be an issue is if you have one of those samples that were sequenced at greater depth, it increases the likelihood that one of those inserts might be a lower expressing transcript that they might pick it up versus something that's not sequenced at that range of depth. But we'll be able to normalize about 30% difference among samples, which I've seen a lot worse. So not too bad. Next question is, what do you notice about the quality of the trim sequence data compared to the quality of the raw data? So Lisa or Armin, if you guys want to move them to the next question. So Lisa and Armin, Elwood submitted an answer, but it didn't post. Is there a reason?
I think it may have gone to the question before because the first question was so long, it was actually split up into two, but we didn't stop. So if you go to your results, it may have
So Elwood, let's see, maybe yours
been
was
for the previous one. I noticed there were two answers.
yeah. So if you did the range of samples just discussed. Yeah, so make sure, guys, because some of these had to be split between two questions if they were long. So just do a refresh, make sure that the question on top, it matches the question that's here. If not, let us know or hit refresh and it should work.
I'll also be faster about skipping those. And let me just copy the link for you guys. Instead of the code, just follow that link that I just sent.
Yeah, so you guys should all see the question now. What do you notice about the quality of the trim sequence data compared with the raw? So for these, I like to look at the quality trimming graphs. I also like to look at the adapter contamination plots in the multi-QC to notice what the difference is in the quality. How many answers do we have, Lisa?
5,
Okay.
6.
I guess we could focus on it. This one does specifically ask about the quality. The next one is adapters. So we have a few more seconds for this one before we go over the answers.
We have 9. Go on.
Okay, let's go ahead and take a look. Overall, the quality of trim sequence are better, slightly better, it's better, it's better. Reversal Puerto Rico are much closer in quality. The trim sequence data quality is better. Quality histogram tail is slightly higher. Yeah, no definition, looks about the same, Improvement in the mean quality scores, quality is better. Is it the ends of the reads? Yeah. So it's true that if you just take a look at the quality graph itself, so taking a look at this graph here, which summarizes based on Q score compared to counts, and then also taking a look at this graph here, which is the quality based on position. And then we notice this kind of like uptick here at the end of the read, whereas if you were to compare it to the raw multi-QC data, again, scrolling all the way up back to here, This looks pretty much the same, right? Difficult just by eye to notice any real differences. But where we really notice the difference, a lot of you guys pointed out, is here. So this is the raw data, and you'll see that even especially these reverse reads kind of dip into below this 28, I think that line is a Q-score mark. Whereas if you look at that same graph after doing the quality trimming, way down, we do see now that they're all kind of in the green, even at the end here. And in fact, see that uptick at the 3' end, because that, of course, is the end that we're trimming on. Yeah, so right answer is awesome. A couple more questions, and then we're going to jump back to lecture. Number 4, this should be a quick one, I hope. Do you still see adapters detected? So Lisa, if you can put up that question. If you guys can all answer the question, if you still see adapters detected. This one's pretty straightforward because if there's none, the multi-QC will tell you exactly that.
We have 9.
Awesome. I think we can go ahead and view. Awesome. Everyone says no, which is absolutely right. So yeah, right here is where you'd find that information in the multi-QC report. And so if there are no adapters detected, rather than graphing it out, it just prints the statement here that tells you that no samples were found with any adapter contamination greater than 0.1% of total reads. So at least there are none greater than 0.1% total reads, which is great. It's one of the purposes of doing the trimming. And so the last question in this section is, do you think we're ready to align the trim reads to the reference genome now, and why or why not? So I'll give you guys about a minute or so to answer this question. Lisa, if you could pull it up. How are we doing on responses?
About halfway there.
Are we ready to align these trimmed reads to the reference genome? Why or why not?
Okay.
Awesome. Yes, high quality and no adapters. No, need to normalize. Can't do differential analysis. True, but we're not on the differential analysis part. Now we're just taking our reads and we're trying to find out where they came from. So you are right that we do still need to normalize. However, that would come after we actually know where these sequences came from. Yes, 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 or at least I hope. Yes, we've taken reasonable steps to prepare the data. Yes, reads have been trimmed and filtered for length, so we can move forward with aligning to the reference genome. Yes, overall seq quality meets criteria for high quality. Additional trimming may start to lose information and content. That's true. You don't want to over trim. Yes, no other processing steps are necessary, assuming you have the reference genome properly prepared. Good point. Yes, as both reverse and forward reads have not been sequenced. There's no presence of adapters, and all have been sequenced, and all have high quality enough. Yeah. So yeah, you guys are right on it. We have super high quality data now. We don't have any adapters anymore. So we're now ready to find out where these reads came from. And so before we get into that, let's talk about, we're going to hop back to the lecture and we're going to talk about the background of alignment. So I first want to make sure, Ty, are you back with us in the main group?
Yep.
Awesome. Okay, so we're going to move on to the lecture now and pick up where we left off in the lecture. And we have about 14 minutes until our break. So now that we assessed the quality of our raw reads, we identified some low quality base calls in there. We also identified adapter contamination. And so we went ahead and did and trimmed and filtered our raw data, and then ran QC analysis again, and we did successfully enhance the quality of our reads after trimming, and also were able to remove all of those adapters. So all the things that we added on artificially that could interfere with the alignment, we went ahead and removed. So now these data look good, and so we can move on to the next step in the pipeline, which is to align our reads. Before we align our reads, as was mentioned in one of your guys' response to the last question, we do need to create a reference. So we need to make sure that our reference is there and formatted properly for the tool that we're working with, which is the STAR tool. So we're going to talk a little bit about that. So basically we have millions of these short sequences. Now how do we figure out where they all came from? Luckily, the human genome, as well as the genomes of several model organisms, have been discovered, and these reference genomes are available in public databases. So this means we have a place to start looking. We essentially have a map, so that way we can try to figure out where these short sequences came from. So there's like different flavors of alignment or mapping. So usually when people refer to mapping, they don't necessarily refer to something as stringent as alignment, looking at base pair by base pair. So you can also map just by looking at the complexity of the read and the complexity of the respective reference. And based on the compatibility of those two complexities, you can get an idea of where things came from. And there are tools that work that way. And so the whole point of this is to tell you where those short sequences came from in the reference, or what are their genomic coordinates. And so alignment is essentially like fine-tuning of the mapping. And so this will allow us really to get down to the nitty-gritty. This is where you're like looking at the sequence of the read, comparing it to the sequence of the reference, and you're really able to see kind of even some of these nitty-gritty differences, like single base pair differences and stuff like that. So alignment takes longer than doing mapping because it's more detailed and has to account for things like insertions, deletions, and stuff like that, which we're going to get into. Yeah, so aligning reads to a reference, we can identify things like those single nucleotide differences, gaps, insertions, in addition to, of course, the genomic coordinates of origin, or where our sequences came from. So there's a variety of different types of aligners, just putting them in kind of these three general categories initially. So there are global aligners, and I have in parentheses the algorithms that are used to perform the alignment. For those of you guys that are interested, this uses a Needleman Wunsch, I'm sure I pronounced that wrong, global aligner. And that attempts to align the whole provided sequence end to end of both the query and the subject or target. So an example of this would be like if you're aligning two pseudomonad genomes and kind of trying to identify single nucleotide differences between two pseudomonad sequences that you have. Another type that would use global aligners is like if you're trying to identify differences between the mouse and human transcriptome, so you do global aligning of both of those. There's also these things called local aligners, and they use the Smith-Waterman algorithm. And this essentially attempts to find hits or chains of hits within each provided sequence. So an example of this would be like identifying a gene that shares a common domain with a target gene. So it doesn't necessarily match the whole thing end to end, but pieces of it match up. And then there's local aligners. So these kind of combine global and local. And these were used initially in short read aligners, and it assumed that the whole read came from one location within the reference, and thus global with respect to the read and local with respect to the reference. Are there any issues with that approach for looking at short reads and trying to figure out where they came from? Any issues if you assume that the whole read came from one location on the reference, especially if it's a reference genome. You guys put in the chat, unmute your mics. What are some issues with this approach? If dealing with RNA and if dealing with those gross eukaryotes, yes. Mike's always hating on the eukaryotes. Yeah, both true. Anyone tell me why there could be an issue? Gene duplication? What do you mean? Repeated genetic sequences. So what does that have to do with assuming that the whole read came from, oh, you're saying because it could come from multiple regions. That's true, yeah. Yeah. But what about just assuming that the whole read comes from only one location on the genome, even if it has the same sequence mapped in multiple locations? Yeah, it could map to the wrong location. Why? Splicing, yes. Yes, that's kind of what I'm looking for. Splicing, sequence is too short. Yeah, that's true, Susie. I am getting into something a little bit different here, more similar to what Joel mentioned with the splicing, but also even more than that, if we're taking a read that was derived from transcripts, right? So remember, transcripts has alternative splicing, like Joel mentioned. It also, there's introns and exons in your pre-RNA. So if your read came from a message RNA, for instance, those introns would have been spliced out. And then if it spans an exon and you're lining back to your reference genome, if you assume that that read came from one location, that might not be the case if it spans a splice site. So we'll get into that a little bit more so that hopefully it'll become clear. And that is it. What if a whole read comes from multiple locations within the reference, i.e. spans a splice junction? And so most aligners commonly used today are local with respect to both the read and the reference because of this issue. So that allows them to ignore poor quality alignment and low quality read ends and adapter sequence. It also allows them to account for read spanning splice sites. And then just a little pictorial representation here of what a global alignment looks like. So again, you're looking at aligning two sequences end to end. And then a local alignment, you're looking at part of one sequence and if it maps to another part of a target sequence. And so just to get into a little more about reads that span splice sites. And so just to remind you guys, we're starting in our original RNA when we extract it. Some of those transcripts are going to be mRNA, like in this example here. And so remember, early in library prep, we're cutting up those transcripts. So again, some of those transcripts could be mRNA that we're cutting up randomly. And so some of those cuts could happen to be spanning splice sites. And what I mean by that is keep in mind that this mRNA was derived from a pre-mRNA, which originally had introns and exons in it. And ultimately that was transcribed from DNA, which has additional like intergenic regions and things like that to consider. And so when we have a read, like this one depicted here in black, that spans a splice site, so a junction between two exons, and then we go and align it back to a reference genome, well, part of it's going to map here and part of it's going to map here. So we really can't assume that that full read just came from one location in a reference genome, right? Because it could span a splice site. And so we have to consider that there might be splice junctions that are separating out the read. So that's why we have to align these locally with both respect to the read and the reference. Again, it says, if we align reads back to a reference genome and DNA, how do we know where an mRNA-derived read came from when it spans a splice site? And so to account for this, there's different types, splitting up aligners again. So there's aligners that are splice unaware, and there could also be splice aware aligners. And so just to remind you what I showed you on the previous slide, we have this black read here that's spanning this splice site between exons 1 and 2 in this example. And so the issue here is when we go to align this read back to the reference genome, we would be unsure if we should map it back to exon 1 or exon 2 or both, and depending on what type of aligner that we're using. So just a little bit about these kind of aligners. So splice unaware aligners are going to be unable to properly align reads that span splice junctions, and thus they're more commonly used for DNA-DNA alignment. So it's people don't use splice unaware aligners anymore. Really, in RNA data, when you're referring to a reference genome. However, could these be used for aligning RNA-seq data? Can you think of a way that splice unaware aligners could actually be used to evaluate RNA-seq data? Would that be possible? Or would it make sense to do? And if so, in what context? To use a splice unaware aligner. Guesses? Could they be used for aligning RNA-seq data? Yes, no, no, yes. If yes, how? If your areas of interest are intergenic, okay, to allow a gap. So remember, splice unaware aligners won't recognize gaps. So if you have references, introns, yes, exactly, Rachel. So if your reference has introns removed, so basically if you align back to a transcriptome, so if you have your reads that came from transcripts, so that came from RNA, and you used an RNA reference or a transcriptome reference that you align back to rather than a DNA reference. And so again, depending on how comprehensive the reference transcriptome you use is, you could have a reference transcriptome that's just mRNA, if that's what you want to look at. You could have a reference transcriptome that contains, in addition to mRNA, also pre-RNAs and microRNAs and long non-coding RNAs. Depends on how comprehensive the reference transcriptome that you have is. So there are these things called pseudo aligners, and what they do, kind of what I was getting at with the mapping, is they compare read k-mers or overlapping subsequences to a transcriptome de Bruijn graph to find transcripts compatible with the read. So they basically take the reference transcriptome, they break it up into k-mer chunks, and then they look at the number of mers, so like 2 mer, 4 mer, 5 mers, depending on the size of your chunks, and they make this graph of how complex it is based on the k-mer breakup, and then they use that as the reference, and then they take your read and do the same thing, kind of break it up into k-mers and look at the complexity of the different 2 mer, 3 mer, 4 mer combinations, and then map it to this transcriptome graph to kind of see, based on that complexity, what is it most likely to be. The reason that this could work is because, or this does work actually really well with, I didn't have that there. This does work really well with higher expressing transcripts. Lower expressing transcripts, though, it has a harder time doing that. The reason that people would do this is because it's a lot less computationally intensive. So when you're doing your local alignment with a local aligner that's splice aware, takes a lot of computational resources. Pseudo aligners that do this mapping kind of trick, just look at the complexity of the transcript versus complexity of your reads, doesn't take as much computational resources and can be done a lot faster. And it's actually pretty accurate. We were looking into this when we were developing our pipeline and where the issue really comes in. And also you could view this in the literature. They've been compared a lot to actual aligners, these pseudo aligners. And the speed is a lot faster in your pseudo aligner. But again, where you really lose that fine tuning is in those low expressing transcripts. So although you can use these splice unaware aligners, again, if you're using a reference transcriptome, of course, not all reference transcriptomes are going to be as comprehensive. Some of them might not contain all of the different isoforms that could exist for transcripts. You might be missing if there's insertions or deletions that could happen in your samples compared with the reference, you wouldn't be able to identify those. So for these reasons, if you really want kind of fine tune to get more accurate with your alignment, you'd want to use a splice aware aligner, which uses the Smith-Waterman algorithm. So again, local with respect to both the read and the reference. And those are going to be equipped to handle intron size gaps, which will improve alignment of reads that span those splice junctions when you align back to a reference genome. And so those are the ones that are usually used for transcript-derived cDNA, DNA alignment, which is what we're doing here. Yes, it is 9:31, so we are about a minute into our break hour, 9:32 now. So I'm gonna let you guys all take the rest of your break time. Please be back here in 18 minutes at 9:50, and we will pick up with these splice-aware aligners.